So I feel very privileged to have been selected as an Nuffield Scholar, particularly when I decided I was going to study soil biology in what turned out to be the International UN Year of Soils. So making the most of your soil biology is what I'm trying to do. I really need to thank Nuffield for selecting a scholar from a very conventional industry that was suggesting a rather, well, at this point in time, considered an unconventional approach. So I thank you for that. I also thank you, Nuffield, for actually giving me the opportunity, because I didn't realise initially that we had to do this presentation twice, but by doing that, they've actually given me a chance to give a 30-minute presentation instead of 15. Farming in the next green revolution was mentioned this morning, and I certainly believe that biology is what will be a very key part of the next green revolution. I also need to thank my sponsor, SRA, Sugar Research Australia. Family farm, fairly small one, near Mackay, and our operation is of the size that it's very difficult to employ labour. So we rely on the family unit, which is basically my parents that are both in their 80s. Dad had a double knee replacement there a couple of years ago, and he hasn't looked back. It has added 20 years to his life. So yeah, he's my number one worker. I also need to thank my dear wife Susie. You are my rock. Thank you. So the reason why, well, Mackay area in particular has suffered a severe decline in yield productivity across the area that's been, as you can see, ongoing now for over 2 decades. There will be many that will argue with me that that's because of a number of different causes at Mackay, and that soil health is perhaps just one of the minor ones. I would dispute that fact and think that soil health is probably the major one. But it's the one that's the most hidden. It's not as easily seen, particularly when you're talking about soil biology. So, can we reverse this by building soil health through biological activity? I certainly fully believe so. Now, we need to, when we're thinking about soil biology, keep in mind that it very much has a symbiotic relationship with plants. So it doesn't matter what sort of soil biology you talk about, I firmly believe that there is a symbiosis with a certain range of plants. So we already know of some of these, like rhizobia and legumes, and AMF, otherwise known as arbuscular mycorrhizal fungi, has a symbiotic relationship with at least 80% of the plant species on the planet. This is probably the largest symbiotic relationship, and it's probably the most beneficial because it allows the roots of the plant to extend much further than they otherwise could, and bring nutrients, particularly phosphorus and micronutrients and moisture back to the host plant. With old growth forests, they've actually established that AMF has actually enabled roots to extend more than 10 metres beyond where they actually were. And it does that simply by the fact that microbiology is so damn small it can fit into any sort of space, and it just spreads out like a web, so long as it can be nurtured and isn't destroyed. So I actually believe in years to come that they'll realise that all biology has a symbiosis with various different plant species, and if you want a range of biology in your agricultural situation, whatever it is, you will need a range of plants, otherwise you'll just limit your biology. I met many inspirational people along the way, and I can't talk about them all. I just don't have time, even if I do have 30 minutes. So I'll just talk about a few of the real key ones. Gabe Brown, for me, farmer from North Dakota in the US. And if you want to see Gabe a little bit more, he's all over YouTube. There's a very good YouTube clip called Soil Carbon Cowboys that goes a long way to explaining his system and shows you a good example of it. And it just so happens I've made my own YouTube clip too, if you want to have a look at that. There will be another one of those later this year. He was a very conventional farmer 15 years ago, and then his soil carbon started out in its natural state at 5 or 6%, and through very conventional practices it declined to the 2% range, and then coupled with drought and high input costs, he just reached the point where if he didn't make a change, he was going to have to sell it. So he went whole hog pretty quickly actually, and made a very significant change with multi-species cropping, and has turned his soil carbon around. So over the last 10 years or so, he's managed to claw his way back to that 5 to 6% range. So anyone that would tell you that soil carbon is impossible to build, they're pulling your leg, because he's a living proof that it is possible to build. Jay Furfer, Research Sciences with the USDA Agricultural Research Service, he actually gave me a very practical idea on how to actually do this. So he led a research project that's now over 10 years old, where they multi-species intercrop with corn. They planted corn on 30-inch rows and then planted the same field the same day, just moved over 15 inches with a multi-species intercrop. So then you had alternating rows 15 inches apart. And they found after 3 years that they had this yield penalty because of what you would all expect, because of the competition effect. And he was a bit worried about it. And then year 4 came along. And year 4 was a horrendous drought. It was their driest year in a long, long time. And guess what? The intercrop section actually out-yielded the conventionally treated section of the trial by 40%, and it's never looked back. It's out-yielded every year since. So how did it do that? By improving soil health on a whole range of levels, not just by increasing soil carbon, but biology can influence everything you have to do with agriculture. I'll just digress a little bit here. I firmly believe that one of the biggest differences between the really resilient, successful farmers and the rest is their ability to take full advantage of the 4 key elements that as farmers we all get that are for free. So that's your sunlight, your rainfall, carbon and nitrogen. And all these things will fall over your soil, and your ability to take full advantage of those 4 things is what sets you apart. And you need soil biology to do it. Without a fully biologically active system, you will not take advantage of those 4 key elements. Christine Nichols, who's the lady in the photo here, microbiologist. She was with the USDA as well. That stage has now gone over to the Rotational Institute. She demonstrated to me how she's actually been working in the area of trying to work out what biology actually is. So calling them different names, determining which one you actually have, and through just magnification basically and some very clever camera work, has been able to come up with a system that's a lot better at actually finding out what biology is in a particular soil sample. The shed that they're standing in front of there is actually to do with rainfall control. So they can roll that shed backwards and forwards, and they can put on 100 millimetres an hour if they wanted to, or 10, or nothing. So they can treat that strip of soil there however they like, because it's big enough to work machinery on, any sort of normal machinery that we would use. So it's more like a proper field trial instead of a pot trial if you want. So they can really control moisture. And when it comes to moisture, that's one of the biggest influencing things. Moisture has the biggest influence on soil biology. So if you want to test for soil biology, you need to pay careful attention to how much moisture you got in the soil, because that will influence dramatically what sort of result you may get on any sort of microbiological test you will do. Professor Stephen Ratton. This photo, he's from New Zealand actually, Lincoln University in New Zealand. And I went to visit Stephen over there, and I was very chuffed when he rang me and asked to come and see me. And this photo was in a sugarcane field at home at Mackay. Professor of ecology, and he's developed, along with another professor from Australia, a fellow by the name of Jeff Gurr, what they term ecological engineering. So that's just basically, to keep it simple, the growing of plants in association to take full advantage of the pest-prey relationship of insects. So the same thing happens with soil biology. There's a war going on down there, and it's very much a case of trying to get a feed, and there's plenty of biology that likes to eat it, as well as there's plenty of insects that like to eat each other. So you need to think about the different plant species you can grow in association to maximise that benefit. Stuart Card, also in New Zealand, has developed epicloe or endophyte, not developed it as such, but has worked out ways to take advantage of epicloe endophyte. Endophyte just means inside, so this is a biology that lives inside plants. With the epicloe endophyte, it was all about ryegrass and controlling things like staggers and sheep. They also found another particular endophyte that insects don't like at all. So they inserted that particular endophyte in ryegrass, and it's now a product called Avenex, and it's planted at airfields all over New Zealand and now all over the world, because insects don't like the endophyte that's in this plant. They don't bother coming to eat it. So if you don't have insects, you don't have birds, no bird strikes at airports. Brazil, well, Chile and Peru and Brazil I also went to. The photograph here is of Denizar Bolisi, Sao Paulo State, and Denizar's been working on a project for quite some time now, all about rotational cropping for sugarcane. So all sorts of different crops they could grow in rotation with sugarcane. But not only rotation, they've been doing a fair bit of work on intercropping as well. But usually just single species to intercrop with the sugarcane. In the photo here, he's actually holding some crotalaria, or what we would call sun hemp. There's many different crotalaриas, and there's some native crotalaриas to Australia. There's also plenty of natives in Brazil, and they have a much better range of crotalaria than we do. And certainly the crotalaria, being a legume, holds some real good prospects for rotation with sugarcane. This, believe it or not, is sorghum, and it was 6 metres tall. I just could not believe how the damn stuff was standing up. Because if it was sugarcane, it'd be lying all flat all over the ground. This is more of Denizar's work, and that sorghum's been developed for the cellulose ethanol industry, because the Brazilians have a very good ethanol industry in conjunction with their sugar industry, something that we don't have, but yeah, perhaps it'll come. Whether or not the Queensland government actually is successful in mandating ethanol, we'll wait and see. I won't hold my breath. Measuring carbon sequestration. It's difficult, but in Nebraska I came across this trial that's been running for a long time now. It's certainly, I think, one of the longest running trials for carbon sequestration in the world. And it's got a further 7 years of funding, so I find that very exciting. They have some very sophisticated equipment here and can measure the inflow and outflow of carbon dioxide on a per second basis. They have three plots in this trial, and the only one of those plots that's actually sequestered carbon over the last 14 years is the plot that contains a perennial. So all three of the plots have corn and soybeans in rotation, with one of these plots having that alfalfa, that perennial, in that rotation. And as I said, it's the only one that's managed to sequester carbon over that period of time. So key outcomes, plant diversity. It's just all about plant diversity as far as I'm concerned. plant diversity gives you that biological diversity. And once you've got biological diversity, you will have a healthy soil. There you have it. For me, the three key things. You need to do all three. You can't just do one or two of them to make inroads in this area. Australia, a lot of Australia is pretty good at doing the first two, except the sugar industry. But they don't do the last one very well at all. You need to do all three of them. The photo in the centre there is just a demonstration of some soybean trash. It's on the farm at home, and I actually direct drilled sugarcane into that. So no-till sugarcane basically, which is, it has been done before, but up until now, not terribly successfully. Well, so far it looks damn good at home this year. And some multi-species intercropping. So I'm setting out to change the paradigm. There'll be plenty of different ways you can tackle this. And this is just my head-on approach. So a full-on multi-species intercrop, 2 rows, as you can see, of literally 8 different species of plants in between the sugarcane. You can barely see the sugarcane there. It's very poor in its early growth at taking advantage of sunlight. Very poor. It just doesn't have the leaf area typical of perennials, whereas your annuals are much quicker and can take advantage of that photosynthetic potential. But I realise intercropping like that, as you can see from the photos, looks pretty ordinary. A lot of growers would just cringe and turn the other way. So I'm trying to drag the industry with me, give them an idea of how they can do this and actually make a profit off it in the early stages. So what we did here is some same field as you saw before with the soybean trash. That's the sugarcane in the centre that my father's standing above with sunflowers on either side. I'm getting the wind up here. So there you have it, recommendations to industry. We really need to take a whole of system approach. You can't just do little bits and pieces of it. I really believe that you can't afford not to seriously consider plant diversity. And I don't think you can apply biology from a drum unless you have the suitable plant species to go with it. My contact details. Thank you.